So hi, my name's Vicky. I'm a second year PhD student at the University of Bath, and today I'm going to give you a quick overview of my project, defining the role of efflux in bacterial biofilm formation and antimicrobial resistance to develop new treatments for infection. Or, for a slightly less wordy and more specific title, my alternative, having a urinary tract infection is quite rubbish, maybe we could find some better treatments. So I'm sure everyone is aware to some extent of how efflux pumps contribute to resistance through the export of antimicrobials. And this on the left just shows us those 5 main classes of efflux transporters that you'll probably all be familiar with. But it's not just drug efflux we've got to worry about. It's thought there's some other roles during biofilm formation, including the secretion of substances which make up that extracellular matrix encompassing the biofilm community, and the secretion of things like inducer molecules and involvement in quorum sensing. So for my project, I'm using Proteus mirabilis as a model organism to look at the role of efflux in UTIs, focusing in particular on catheter-associated UTIs or CORTIs. So this catheter here, obviously ideal surface for bacteria to grow on, bypasses body's natural defences, makes the likelihood of developing a UTI much more increased, as is the risk of severe complications. And Proteus is the cause of just under 50% of all catheter-associated UTIs, and is the most likely reason for catheters to block, due to its ability to form this lovely crystalline biofilm incrustation, which you can see here. So Proteus produces urease, which rapidly raises the pH of the urine. And this causes precipitation of things such as calcium and magnesium ions that are found naturally. So you can see the complete incrustation of the catheter tip here. So that's this little bit. So where this balloon is, this will be inserted with the balloon deflated, then that balloon is inflated, sits in the bladder to stop it leaking out, stop it falling out. Then you've got the little drainage holes at the top here that the urine can drain through. So if you've got this complete incrustation, clearly no urine is going to be draining through there. This is going to lead to urine retention, backflow of infected urine, and therefore increased likelihood of the complications. So to try and reduce this happening clinically, sometimes things like biocides and chlorhexidine is used to try and reduce this biofilm formation by physically flushing through catheters, and as well as for extensive cleaning of surfaces in hospitals, care homes, and nowadays pretty much everywhere, obviously due to increased infection control measures. So there's a lot of evidence now that there's increasing tolerance of Proteus to the use of these biocides, in part due to the upregulation of certain efflux systems. And this increased tolerance to biocides is often associated with cross-resistance to antimicrobials. So inhibition of efflux systems could therefore be an important tool for both inhibition of biofilm formation and increasing the susceptibility to antimicrobials. So due to this blocking ability, we can use Proteus as a model in this in vitro system. So this is our glass bladder here with the catheter inserted up, and you can just about see that inflated balloon that's holding it in place. So we can use this model as blockage time and pH changes are good measurable endpoints to compare different treatments or to get an idea of the behaviour of different knockout mutants that we've created. So this one on the left is a control with no inoculation, so you can see nice and clear, good drainage through. This one is an infected model, so it's not yet blocked. You are still getting the urine draining, so you can see this is the level of the urine here. But obviously, this is quite an infection at this point, quite a lot of biofilm buildup. And before too long, you're going to get this incrustation that you can see here, complete blockage, as you can see the level of the urine sort of representing this retention that would effectively be taking place at this point. So obviously in a patient, this is not an ideal situation. So previous work in the group has started to investigate the role of certain efflux systems and how they might be linked to biofilm formation and resistance. So transposal mutagenesis approach was used to identify mutants with inhibited biofilm forming abilities. And through this process, one of these mutants identified that the BCR-CFLA MFAS efflux system was inhibited. So knockout mutants were then produced, put through the in vitro bladder model system. This caused a significant increase in the time to blockage, reflecting that reduced biofilm forming ability of that knockout mutant. There was also increased susceptibility of isolates to certain antimicrobials. So following on from the success in the bladder models, this was then selected for in silico molecular docking simulations to try and identify inhibitors of this BCR-CFLA system from a database of existing drug compounds. So this identified that fluoxetine and thioridazine were able to reduce biofilm formation when they were added to these catheter models in an infection simulation. So you can see here there's a dose-dependent increase in blockage time. So this longer blockage time is reflecting the fact that it's taking longer for that biofilm to form, and these are obviously slowing down the rate of this formation on the catheter. compared to the untreated control. And this was also repeated in other urinary pathogens, so E. coli and Pseudomonas aeruginosa, and to a lesser extent, but there was still a significant decrease in the amount of biomass formed. You can't measure the blockage time for these, so you just purely measure the biomass formed over a certain period of time, and it showed that this is potentially going to be able to be reflected in other species as well, not just Proteus. So it's not quite as simple as just pick an efflux system to inhibit and decrease the biofilm formation. So we do need to be able to understand more about the role of different systems and able to choose appropriate targets and then develop inhibitors for them. So following on from that, I've got two main aims overall, which is to investigate which efflux pumps have a waste management and regulatory role in biofilm formation, and also whether drugs already used in human medicine could be repurposed as efflux inhibitors and antibiofilm agents. So this is just an overview of my overall plan. So firstly, I'm going to look at characterizing efflux system genes across sequence Proteus mirabilis isolates. Secondly, using RNA sequencing to look at the actual expression of these efflux systems under different conditions. And then using information gained from these first two stages, choose some key efflux systems to generate knockout mutants for, and then obviously see how this changes their sort of phenotypic characterization and also their virulence by using the Manduka-Sexton model. And then finally look at the use of in silico modeling to assess interactions between target systems and their potential inhibitors. So firstly, I've used the genome sequence of the most well-characterized Proteus strain, HI4320, as a reference. And I've identified all efflux systems and their substrate profiles and associated regulators where possible. So this initially was isolated from a urinary catheter. And you can see here, I've just picked out I've just grouped them by class. We've got the 5 main classes here. I've also picked out the 6th class PACE, which is involved in biocide and quaternary ammonium compound efflux. This other category just includes any other system that I could identify that had an energy-dependent efflux function. So following on from this, I then found all the available genome sequences I could online, and using this HI4320 as a reference, I sort of made a variant database basically of all of these 551 sequences by doing variant calling against the reference, and just created this database where I could go in and look at individual genes and compare the variation of these efflux related and regulatory genes to the genome as a whole and to each other. Also, since being able to get in the lab, I've got an additional 50 clinical isolates that we've got as a panel that I'm hoping when my sequences come back, I can add to this database as well. So I've done various phenotypic characterizations of the clinical isolates so far. And like I said, when I'm getting the genome sequences back, hopefully we'll be looking at the efflux genes to see if this matches any potential changes. So I've done various things, so just basic growth and biofilm formation screens, some MICs, and bladder model blockage times for those strains which came up as being highest or lowest biofilm formers in that initial screen. So of all the strains, this one, RS42, was the most impressive biofilm former. So this was it in the bladder model. So this has got quite a significantly shorter time to blockage, so about 12, 13 hours compared to that HI4320 reference, and also compared to most of the other strains that we put through. So I also did MICs, and it had the highest chlorhexidine and polymyxin MICs. At the other end of the spectrum, we've got RS33, which was much more susceptible. So Proteus is considered to be intrinsically resistant to polymyxin. So actually to have an MIC of 32 micrograms per mil, that is actually quite a susceptible isolate. So that's quite interesting in that respect as well. So based on the characteristics of these two, I've chosen these to go forward, and I've done some RNA extractions for both of these strains, both with and without exposure to chlorhexidine. I've also done extractions from the planktonic cells, which I isolated from the artificial urine in the bladder model. So when this data is back, I'm hoping that, again, this will give me some clues as to the relative importance of different systems under different conditions. I'm also currently trying to work on getting RNA from the biofilm cells on the catheter itself. This is proving slightly tricky due to the fact that it's obviously quite calcified, so not the easiest to extract. But I'm hoping that at least by looking at the expression in the planktonic cells, this may be to some extent representative of at least what is sloughing off the biofilm surface of these sort of biofilm associated cells. So hopefully there's going to be much more exciting data still to come. So like I said, still waiting for quite a lot of genome sequences to come back and the RNA data. Another quite exciting thing that's in progress at the moment is I've carried out a sort of adaptation of HI4320 by doing sort of subsequent passages every 48 hours, exposing it to doubling concentrations of chlorhexidine. I've adapted it to grow at this really high concentration. I took samples of the populations at each stage and obviously at the final stage as well. And then hopefully when we get those back, be able to look at what point different mutations are occurring and see if the mutations are occurring in specific efflux systems. Maybe these are occurring in the wild type sequences in the database as well. So this will be another really interesting thing to look at, and obviously using all of this information to then choose those systems to knock out and then move forwards with the rest of my project. So thank you very much for listening to my very brief, garbled overview of my project. Just to say thank you to all my supervisors and the rest of the Jones Group for their help and support so far. Thank you very much.